Hi there and welcome to this IB revision video for IB Biology, and this contains content for both standard level and higher level for 2023 onwards, and today's video 5 on nucleic acids, and we won't be going into the function of them, more just the structure today. So you need to know that nucleic acids are essential in living organisms, and they are polymers or polynucleotides and contain the information needed to create an organism. So they are monomers of nucleic acids, and they come in two forms, DNA or RNA. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. They hold the code for building the organism, which we'll look at later on, and almost all DNA in eukaryotic cells is found in the nucleus. There's also some in the chloroplasts and mitochondria, which make proteins for those particular organelles, but we're talking generally about the nucleus unless we say otherwise. And there are three types of RNA which read the DNA and translate it to make proteins. And we'll look at this in more detail separately. You need to know then a monomer. So we said the nucleotides are polymers of nucleic acids. The monomer of a nucleic acid is a nucleotide. So a nucleotide is one individual part of the DNA molecule, and you need to be able to sort of draw this structure you can see here, and you need to be able to describe this structure as well. So you need to be able to say that it is made up of a phosphate group. That phosphate group is then attached by a covalent bond, which we'll name shortly, into the pentose sugar, so it's a 5-carbon sugar, and that is attached then to a nitrogenous base, and the nitrogenous bases obviously all contain nitrogen, and there are three names of those. So just to point out, the pentose sugar is either deoxyribose in DNA or ribose in RNA. So there are basically five different types of nucleotides. You could say 8, depending on how you look at it. These nucleotides then are the nucleotides for DNA, okay, and these nucleotides then are the nucleotides for RNA. And you can see here there are 4 organic bases on each of them: A, C, G, and T. Whereas this one is the same again, ACG, but this time it's U, where something called uracil replaces thymine. And again, notice the difference here. We've got deoxyribose as the sugar and ribose as the sugar in RNA. So you need to know that the phosphate group is always the same. The sugar is either deoxyribose or ribose, and there are 5 different bases: adenine, thymine, guanine, cytosine, and uracil. And basically, thymine is the organic base in DNA, which is replaced by a very similar molecule, uracil, in RNA. So in terms of making this into a longer polypeptide chain of nucleic acids, we need to have these nucleotides obviously connecting. So what we will have is a reaction happening, forming this bond between the phosphate of one molecule and the sugar of the next one. Hopefully only molecules of the same sugar can join, so DNA can only join with DNA, not with RNA. Now there is something which you hopefully have probably already picked up on, that there is a complementary base pair ruling, and this is something called Chargaff's rule. And basically the complementary base pairs are A and T, so adenine always combines with thymine, and that can be replaced by uracil if it's DNA and RNA. G always combines with C. Now you also need to know the number of hydrogen bonds between the two. So between A and T, they're held together by 2 hydrogen bonds, whereas G and C are held together by 3 hydrogen bonds. And that's just because they're complementary, so they interact with each other. Now this base pairing, or this complementary base pairing, is universal, meaning all living things follow this rule, because we have a universal genetic code, so we all use DNA and RNA. Also notice that in this chain or this strand of the two-stranded molecule, that the molecules are pointing upwards, and in this strand they are pointing downwards. So that is called they are anti-parallel to each other. Now there's a little bit here says about this which I'll come on to with a higher level only, but just be aware that the distance is always the same. So this distance between here and here, this width of the chain, is the same here as it would be 100 bases further down. It's exactly the same, which adds to the stability of the molecule. So this is a bit I was mentioning, and we will be doing some more standard level stuff after this, so don't stop listening if you're a standard level student, but you can skip this one slide. So when we said this A to T, G to C, what we're also saying is that a purine and a pyrimidine combine with each other. So the two molecules, if you look at this diagram at the top here, C and T, are a lot smaller than G and A. And that means C and T and U as well, don't forget, because U replaces T in RNA, are called pyrimidines. And pyrimidines are smaller, they have a 1 carbon ring structure. Okay, so they've only got 1 carbon ring, one there, one there, one there. Whereas A and G are purines, and they have 2 carbon rings all the time that make their structure bigger. So if you look at that and go back to what I said a few minutes ago about the width of the DNA molecule, it is always going to have a width of 3 carbon rings in length, because you've always got 1 purine, 1 pyrimidine together. So that gives it an equal width, and that width is 2 nanometers at all points. So that means that it's consistently strong 3D shape, and with all the hydrogen bonds connecting each base, that means it is an extremely stable molecule, which is essential for DNA because it's information storage. So you need to know they come back like a ladder. Like the phosphate sugar backbone is what we call the uprights, and the bases would be the rungs of the ladder. Hydrogen bonding makes them very stable structure, and the phosphodiester bond, which is a covalent bond, which we'll talk about in a few moments, is extremely strong as well, holding together the one nucleotide to the next nucleotide. And it is vital, because if we have an unstable molecule, we'd have more errors, more mutation errors. That would lead to more mutations, and that would generally lead to problems in hereditary from organisms. So let's just look at the phosphodiester bond a little bit. You need to understand exactly the same as before. It's unlikely you're going to be asked to draw a diagram to show this one, but just to show you, it is again a condensation reaction. Okay, forming a phosphodiester bond. Don't call it a covalent bond, although it is a covalent bond and it's strong as a result. Okay, water is removed by reacting, and this is the detail you need to be able to say, the hydroxyl group on carbon 3 of the ribose sugar. So what we mean here is this hydroxyl group, we're reacting that hydroxyl group with the hydroxyl group of the phosphate here. Okay, we take out a water molecule, leaving an oxygen as before. There's your oxygen there, and that then creates the phosphodiester bond, and that again is a condensation reaction. Now, the structure of DNA is essential because it is the information to make proteins. Molecules are long, so they can have huge amounts of information, and we have base pairing rules, which means it's complementary across all the DNA strands in the world, and therefore we can replicate it. And it is double helix, which gives the molecule stability, and our hydrogen bonds both give stability, but also allow the unbinding. Now, we won't go into this now, but just to inform you, this bit here, we'll often talk about the term genes when it comes to DNA. A gene is a sequence of DNA bases. By that we mean the letters, the code that code for either a polypeptide or a functional RNA molecule. So functional RNA is something like tRNA, which we'll talk about later, which allows the creation of proteins. And what do you need to know? Well, you need to know every 3 bases is called a codon. It may also be called a triplet, but we'll stick with codon. Okay, and every 3 bases therefore is then copied by RNA. Okay, so and then that RNA molecule will then do something called translation, and it will make one amino acid. Don't worry if you don't fully understand that yet, because it's not on this video, but it will come up later. So if you just remember, every 3 bases is called a codon, and every 3 bases codes for one amino acid to make the primary structure of protein, which we covered in the previous video. Now for HL students, this is now going to be quite important, and the rest of this is now HL, that you have anti-parallel strands and what this means. Now we'll come on to this more again when we do DNA replication, but there are 2 different ends to the amino acid, sorry, the phosphodiester backbone. So this end, we've said earlier on, is effectively pointing up because it's anti-parallel. This one is pointing down. Now, why are they pointing up and down? Well, basically, it's the direction of the sugar molecule. So each sugar molecule has different amounts of carbon. So we've got carbon 1 here, carbon 2, carbon 3, carbon 4, and then carbon 5. So because we have carbon 5 at the top, we call this the 3' end. So there it is there it is there, but this one, notice, is upside down, so it's there. And then we have carbon 3, which we call the 3' end. So we've got the 5' end, sorry, I think I said that wrong a minute ago, and the 3' end. Okay, so we've got carbon 3 down here. Oh, I've almost deleted that, but never mind. Carbon 3 here, and this one, it's going to be the other way around. So we call this anti-parallel because we base it on the direction of the 5' end. Now this is important because an enzyme we're going to talk about when we do about DNA replication, called DNA polymerase, can only attach to the 5' end of the DNA chain. So what that means is the DNA polymerase, which is essential for making new strands of DNA, can only attach effectively here, or on this side can only attach here. Now what that means is it will attach here, so if I use it at the bottom on here, and it would move up the strand. So if it wants to attach new DNA nucleotides over here, it will be basically starting at this end and working its way up. So it can attach the 5' end, but it works on the anti-parallel strand, so it can only attach to the 3' end. So it builds the new strand from the 3' end upwards. That will become more clear when we do DNA replication. So at the minute, I think the key thing you need to know is that carbon 5 is at the very end of the chain. Sorry, I should have said that. The phosphate is unlinked, okay, and at the very end of the 3' end, this bit would be unlinked at the very, very end. And an enzyme that replicates DNA can only attach that 5' end as a result, okay, and it's because it has a complementary shape. So the enzyme essential to making new DNA strands can only attach to here. Imagine it like that, because it's got a complementary shape. And again, down here it could attach because it has that complementary shape. So DNA polymerase can only attach to the 5' end because it has a complementary shape. So the DNA code is what we call a common degenerative non-overlapping code, and it's important you understand those terms. So first of all, just to remind you, this is sort of a DNA wheel here, and it shows you the codes needed for different amino acids to be made. So let's say I wanted to make amino acid serine here. Okay, I'll go from the inside out, the 5' end outwards. So the code for serine would be U, C, and then one of these. So it could be UCU, UCC, UCA, or UCG. Okay, now all amino acid chains always start with something amino acid called methionine, and that would be A, U, G. So the first code in all genes that are going to code for a protein is A, U, G, which makes methionine. So this is called a universal code because the code is the same in all living organisms. It is universal. So if I wanted to make methionine, it would be UCG for me. No, it wouldn't. I've read it wrong. It would be I keep reading it wrong. AUG for me. If a shrimp wants to make it, it would be AUG. The same code for the same amino acids. So it's a common or a universal code. The code is degenerative. So what that means is there is more than one way to skin a cat, or in this case, to code for amino acid. So if we look at alanine, okay, we go G, so they all start with G, C, and then either GCG, GCA, GCZ, or GCU. So there are several messenger RNA codons that code for the same amino acid. This is called being degenerative. And an advantage of this is if you get a mutation, you may still end up with the same amino acid, so it might have no effect, which is a positive. So CCG, if that G got swapped for an A via mutation, it would still code for the same amino acid, alanine. And then we've got then the further bit, which is they are common, non-overlapping. What this means is it always is coded for by 3 bases. So every 3 bases in the amino acid sequence codes for one. In the DNA sequence codes for 1 amino acid, and the ribosomes will read the DNA sequence 1 base or 3 bases or 1 codon at a time. So it never overlaps. So we call it a non-overlapping code, and each base is only read once. So all the latest information is just for HL students, and it takes it to a higher level of what the structure of DNA is within a chromosome. So we're going to start off by looking at the structure of something called a nucleosome. So a nucleosome is what we call a structural unit of eukaryotic DNA chromosomes. It's only for eukaryotic cells. And basically it's what you would think of if you're describing a chromosome. So it's a length of DNA wrapped around or embedded in a core of a histone proteins. Okay, so, and you can see here a diagram image of it. So you could be asked to describe what a nucleosome is, or what the structure of a chromosome is, of which you could then describe this. So basically the nucleosome is 8 histone proteins, and there are 4 types of histone proteins, so you get 2 of the 4 different types. And as you can see in this diagram, the red bit is DNA. So the DNA is wrapped around the histone proteins as a result, and it's wrapped around twice. So imagine this as a spiral, it's going round, behind, round again, and then back this way. There is a H1 histone protein, which is an additional protein, which, if you think of it like a cord, it's like holding it on tightly, and it reinforces the binding between the DNA and the nucleosome, so it keeps it sort of tightly on there. And this can become tighter or looser, and that is useful when you are preparing for DNA division. So if you think about mitosis, which you may or may not have studied yet, mitosis is when the DNA molecule is becomes, or the chromosomes become compact or thicker, or you might use different words for it, but this is because the H1 histone protein tightly packs the nucleosome together. Then there is a short section of DNA, which is pictured here, which will bind then to the next nucleosome, and so on and so on. So chromosomes will be lots of nucleosomes attached to each other. Now bacteria don't have any histones, so they don't have nucleosomes, so they have what we call naked DNA. So we all take it for granted. You've sat and listened to me saying, oh, it makes the amino acids as genetic material, you know it's passed on from parents to their offspring. But actually, why do we know this? Why do we know that DNA is the genetic material? So if we look at the history of this, in the 1800s, scientists believed chromosomes had a role. So we realized you could look under light microscopes and see this very basic use of chromosomes. But it wasn't until the 1900s that we realized that chromosomes are made of both DNA and the histone protein. So in the 1940s, so less than 100 years ago, scientists believed that proteins were the hereditary material. And if you think about it, why this might be, well, proteins are the same in all different living organisms. They've got specific shapes, they've got things like that. But the proteins were effectively made of 20 amino acids compared to 4 bases of DNA. Now, why is this important? People would have thought, well, that gives more variety, so therefore you get more. You would get more the variety of life we have, because variety is the key for hereditary. However, the experiment in the 1950s by Hershey and Chase disprove that proteins were the hereditary material and prove that it is the DNA bases that are key. So Hershey and Chase basically did an experiment where they looked at, is it DNA or is it proteins? And the key thing here is to remember DNA contains phosphorus, but it doesn't contain any sulfur, and protein contains sulfur. So if we're looking here at this experiment, you could see that if we're looking at if sulfur was passed on, it would prove that protein must be the hereditary material. Okay, but if phosphorus is passed on, then it would prove that DNA is the hereditary material. So what they did is they grew viruses called the T2 virus with either radioactively labelled phosphorus, which would then be able to be detected, or radioactively labelled sulfur, so isotopes of the two. Okay, so what did they do? What they did was they used two different experiments, very similar, very simplistic. They used proteins to identify whether it was proteins that was being passed on. They used radioactively labeled sulfur. So the bacteria, the virus's protein coat was labeled effectively. They allowed the virus to infect bacterial cells, which obviously if they infected it, the DNA gets incorporated into that. They then blended this material up, and what they found was that there was radioactively labeled sulfur present in the supernatant when they spun it at high speeds. So they spin it at high speeds, and the bacteria cells would have gone to the bottom because they would have been heaviest, and as a result, if the protein from the virus was incorporated into the bacteria cells, they would have had the protein in this bit here, but it wasn't. It was found in the supernatant. So what this tells us was the radioactivity was in the supernatant. The protein therefore had not been taken up by the bacteria, and it showed that that isn't inherited by the bacteria. The virus doesn't replicate this way. Now, when they did this with phosphorus, it was obviously the DNA that was radioactively labelled. That then did get incorporated into the bacterial DNA, and when they then blended the mixture up and then they spun it at high speeds, the only bit that was radioactively labelled was the pellets at the bottom, which were the bacterial viruses. So this shows us radioactivity was in the pellet, the nucleic acids have been taken up by the bacteria, and it shows us that bacteria was using the DNA to replicate, and thus the viruses were using DNA to replicate, and this then allowed us to understand DNA is used in replication, not proteins.